Hi, I am Ilya Rasin from Robots&Parrots, and today I'm very excited because I'm starting a series of tutorials about neuronal networks. Today we are talking about uniqueness of neuronal networks. Next time our topic will be about similarity of neuronal networks and artificial neuronal networks and real neuronal networks in our brain, in our body. And then we go to mathematical foundation, which will consist of two parts. One part is about so-called dot product. This is the main operation which is happening during the training of neuronal networks. And the second part about partial derivatives and gradient, especially gradient descent. So let's get started. That will be 5 tutorials, and today is the first. Today we will talk about uniqueness of neuronal networks. As you see, the topic of today is understanding of neuronal networks based on human DNA. Why I have chosen this as example? Why human DNA? Because the DNA by itself is also intelligence. It is our body has two main center of intelligence. This is the brain and nervous system, and also every single cell of our body. So our intelligence is distributed among all cells of our body. So human DNA is very, very important. And you will see in during the session that the structure of human DNA by itself is very similar to what is a neuronal network producing. So let's get started. What is actually human DNA? So human DNA is a huge molecule which consists of, it's a double helix, so two spirals, and this molecule consists of so-called nucleotides. Nucleotides are by itself small building blocks, small molecules, and there are several types of it. So altogether in one of this double helix, so in each of the spiral, we have approximately 3.2 billions of these nucleotides. There are 4 types of these nucleotides. As you see, this is A is adenine, G is guanine, T is thymine, and C is cytosine. And they are, as you see here, I will magnify this. So you see that the nucleotide of upper helix, oops, it's too big. I have to select smaller one. G here, or C, I'm not sure because of it's a kind of little bit pixeled. Let's assume it's C. This one belongs to the upper helix, and another one belongs to the lower helix, and they are connected here via so-called hydrogen bound. But it doesn't matter. So this is all those biochemistry. We don't need everything from it as of now, but just for understanding, so they are kind of building blocks, and they are by itself also molecules. So, and they are ordered in a special way. So they are in sequence, as you see here, A, T, C, and so on. So they have special sequence, and this sequence is very, very important. We will see it next time. I mean, not next time, but in the course of this session. So okay, but don't forget what we want. We want to build a small data set out of it, out of this human DNA, to showcase how the neuronal network works. This is the idea. So why I have chosen this example. So now, to be able to train neuronal network, we must be able to assign labels to it, to every single data point of the data set. So we have data set. This is a table, like a simple Excel table or something like this. You can imagine it. And there is data inside of it and also label. This kind of learning is called supervised learning. Why supervised? Because the network does something, assigns label, and we can compare it. There is a program which does this, of course, but it's our supervision. We can see how the network performs. So we have some labels, ground truth, and we force the network to learn how to assign this ground truth. He will try, error, and there is a mechanics inside of it which will teach him to select right label, to assign right label. And here, if we speak about the DNA, how can we build data set out of it? Okay, we can. There are many ways, of course. This is a huge topic. This is the area of bioinformatics. They do it all day long. But what we can do for our small example is following. Let us cut the entire sequence of DNA into smaller pieces of 4. So we cut it into pieces of 4 nucleotides, and every single piece we put into this table, row by row. So we have, we will build kind of table where we put, let me write here. So we will have table with 4 columns, and then we put into this table at every column. So meaning we take this A and put it in the first position, take next A, put into the second position, and so on. But I will explain it on the next slide in details. Now just, preview what we will see in the next slide. So we will take this, guys, put it into table, and assign labels which we get from any bigger laboratory who does genomics. So we need to get entire data from it. We need also to get the DNA data from it. Assume we are collaborating with such laboratory. This is not unusual. I have done this before. And there are laboratories in Europe and in the entire world which are very, very powerful. They have incredible devices, like from 22nd century. And these devices are capable of doing unbelievable things. So they can run full genome sequencing. It is quite accessible. It's got very accessible, actually, almost to everybody. So you just need a little bit cash. It's around $1,000. It's $1,200, something like this. And they do full genome scanning, and they know exactly if there are some problems with particular chunks of this DNA. These chunks, by the way, are called genes. They are not necessarily 4 elements long. They could be quite big. The idea of genes is that the gene is responsible for certain functions in the body. One gene can affect many functions, many functions, or single function. There are differences. But genes are also chunks of DNA which have some function. Okay, we here cut it in chunks of 4. Doesn't matter, is it a gene now, or is it a part of a gene, or whatever. So and we can assign this label. So assume we got everything from lab, DNA data, chunks, and we assign. So 0, we assign 0 for no disease, and 1 of be suspicious for causing some diseases. Example, it can cause really several diseases. And especially I know it from area of blood cancer. There are actually almost every single type of blood cancer is caused by genetic failures. Especially there are some famous, and there are many types of these errors in the sequence which we see here. Known are deletion, for example. So if one of the nucleotides is simply deleted, somehow disappeared from the sequence, or if there are some rearrangements, like if they are swapped, for example, if in this case, sorry, you don't see it, if they are swapped TTAC, so if, for example, T and C are swapped in this particular position, this causes huge problems actually. And there are some famous rearrangements. For example, it's called Philadelphia translocation, which caused a particular type of malignant disease of blood. Okay, so this is the idea. So we take these chunks, assign labels, and put it into dataset. Again, I started to explain this. How do we put it into dataset? We have we specify dataset of 4 columns, simple table, and then we put first under the first position, second under the second in second column, third in the third column, fourth in the fourth column, and here the label. I mean, this is just as example, a very oversimplified example. In real life, it could be everything. It could be much broader, have different things inside, and so on. So, but now here we build such a simple dataset. It would be quite big. So if we have 3.2 billions divided by 4, we would have 800 million rows. It's huge, actually. It's big data already, but doesn't matter. We will figure it out how to make the best out of it now. Okay, so we have created it and written AATC and so on into dataset. But following problem: all machine learning doesn't work directly with categorical data. I cannot write into cat or dog or AATC or whatever and expect from my program to run on it, from my network to train on text. There are many ways. This is a special area in machine learning, what to do with categorical stuff. And we actually have to select something instead of this AATC and so on. What can we select? Actually, there is already something which we are supposed to do, supposed to use. It's called gene expressions. We assume this laboratory was so nice to us and gave us also gene expressions. So not only the sequence of the genes, not only the label, but also how pronounced is certain gene. It's a complex methods to make it visible. They do it via some luminescent colors and so on, but they see how pronounced this gene. So that means following. The genes could be there in the DNA, but the body reads them. So there is a mechanics how they are read and then translated into certain body functions. For example, into protein synthesis. So proteins are our, we are consisting of such molecules which are called proteins. So huge compounds, complicated molecules. So there are cells. Again, there's DNA in cells. This DNA has genes, these nucleotides, and these nucleotides are read, scanned by a special system in our body, and then something is built out of it. So if they are expressed strongly, more pronounced, they are surely scanned fully, and they are executed, and then everything will happen accordingly. If they are a little bit hidden from the scanning mechanism, they are not so expressed, they are not read or kind of avoided or something. But again, short disclaimer, I'm not bioinformatic guy, so I have some knowledge of this area, and I also do very oversimplified presentation of it, because we don't have time to do now exact explanation what is gene expression and how to see it. But again, it is there, and it can be, even if you print it, you will get such curve for gene expression. So it's something like similar to sine curve, but very irregularly. So it's from chunk to chunk, it will have different shape. It's not the same everywhere. It's not a sine curve. So they have different expressions. And so we write these expressions here. Let me correct it, because I have used just random numbers here. 1, 2, 3, 4. It's bullshit. Let me write maybe 1 here, okay, and here 5, and here 3.5, 4.6, something like this. It's not a natural number, of course. It's something, fluctuating strongly. So gene expressions. So we write them into table. We got everything from the lab with which we worked together. The lab was, we signed heavy NDA, of course, for this. We are not allowed to share this, but for the research, or if you work together with this lab and develop such algorithm, they will share everything with you gladly. Okay, so now again, one short remark here is following. We need also the exact position within DNA. So now I have just written, and maybe we can use index IDX, I write here, and then it would be 0, 1, 2, and so on. So we need to know where it is located, because it does make any sense. It does not make any sense if we just write the expression and the position within this chunk without writing where it is, because it's of huge importance. We need exactly to know exact position within DNA, where this chunk starts, and gene expression, and we need also labels. Okay, now, since we have already built this data set, which will look like something like this. So this is the position 1, 2, 3, 4 in chunk. This is the label. And here we have gene expressions, like 5.6, 3.2, 1.2, 4.0, and here 0, and so on and so on. Entire data set. It will be huge, 800 millions. So it's a huge data set. Sorry, you haven't seen this. It's a huge data set, and we want to put this data set into this network. So what is the network? Network is a layered construction. It has several layers. It could have, normally it has not like here, 1, 2, 3. This guy has only 3 layers. This is a tiny, tiny shallow network. It's not deep. So normal networks, if you do some, if you test and run and something, it will have a couple of layers. I don't know. It will not be so deep. It will be 8, maybe 5, 6, 7 layers if you experiment. And for production purposes, so if a big company has huge computer farms, like Google and so on, they train deeper networks. So it can have also 100 layers or whatever of kind of quite complicated architecture. So now we look into very, very simplistic, the most basic neuronal network design. And so we have 3 layers. The first is not a real layer, it's just the input. So input is a doorway. So we have kind of 4 holes. Into these holes, data is floating into our network. It does nothing, actually. So the first layer, second layer, and output layer, they do. So what do they do? We will see. So now we see that the first layer has 3 neurons, only 3. It's really, really tiny. So 3 neurons. The second layer has 2 neurons, and the output layer has only 1 neuron. Why 1? Because our task is to assign label 0 or 1. So in this binary thing, 0 or 1, we don't need more than 1 unit for this. So the output of this layer will be either 0 or 1. And we have a real label, ground truth, and in this way the network trains. So he outputs something and then compares to the real label, and then the whole story starts again. So he tries to adapt, tries to find the right configuration to assign correct label. Okay, this is our composition. So we have only three. They are called hidden because they are behind input layer. They are behind the door, behind the closed door. So they are hidden. You can imagine this like this. So, okay, now we know what we do. Okay, and now comes the magic. I'm going to explain it one by one. So don't look at the bottom, please. Just look at the first line. So first line, here are the original inputs. Here coming the original expressions. So this is from the first column, from the second column, third and fourth. Here are gene expressions. 3.5, 2.5, 6, whatever it is. This guy is black. I write here 7. Okay. Now, what is now happening is the magic. So is the why the networks are so cool. They actually do following. So we have here 3 neurons. These colored things are neurons. 1, 2, 3. All these 4 original data floating into each of this neuron and build unique combination there. So the red one is the unique combination of these 4. The green one is the unique combination, and the light blue one is also a unique combination. They are all different. Now we have actually, out of these 4, we are all of a sudden three different combinations of them. Now I will cover this. Now all these 3, oops, my picture is covering this. All these 3, 1, 2, 3, they all by itself are now unit. They are isolated units. And they, exactly as before, they are floating, they are going into the next layer, into both neurons. There are 2 neurons. This big guy, orange, is 1 neuron, and yellow guy is also 1 neuron. The second one. And so this 3 things, three combinations, they are floating into the orange neuron and build one particular combination, and then they are floating into the yellow neuron and build another combination. So we have a lot of things combined and recombined again. we have combined one time, and we combined second time. So this is combination and again combination of combination. Now what is happening here? The same thing. Oops, got everything red, but not bad. Okay. Now everything here from the first neuron, from the orange neuron and yellow neuron, they both floating into the last neuron, and they both are combined together to this huge thing, which is in blue. So this is one single combination. And this is what the network outputs. Of course, it doesn't output so many numbers. It output only one number. Why? Because this is everything then combined together via so-called affine transformation. It's just a linear, another word for a linear combination. So it is summed and multiplied with weights. But what it uses, the input, it comes from all those transformations before. So this combination, this huge combination, is converted either to one, to 0, or to 1. And then it is compared with the label. If it's correct, then it's all good. If it's not correct, it will run again and again and again, and try to find a right parameter to achieve this 0 or 1, depending on what is the true label. Okay, and this is the idea. So you have actually staged process, and in this stage process, from stage to stage, you combine the raw data to different combinations. A number of combinations depends on number of neurons per layer. That means the more neurons we have, the more combination we will have. The more combinations we will have. And the more layer we have, again, the more combinations it will produce. So it will expand per neuron, per number of layers. And this is, funny. You can compare it with somebody who is preparing a meal or artist. So you have basic components. Let's say meal. You have basic components, and you start. You mix it together, cook it, then you put it aside. First stage done. Then you mix this product. You have already kind of mix. And then this mixes, these parts, which is already kind of half products, you mix together again together, and then again with something, and then you have a complicated meal, like 3-star from 3 stars restaurant. Or as an artist who is creating a sculpture or painting something, you have also several stages in this process. So you have basic components, you have colors, for example, and then you mix it, and then you paint. So you have already several stages. You paint with the mix. And this cook or this artist has created a picture, and then it gives it a name. And in our case, this is the label. So the network assigns the label. Okay, we don't. The artist has not to be trained to give names to his artwork. This is something else. But the process of creation is very, very similar. It's very similar. It's almost the same, actually. So, and now, it's kind of interesting thing. So if you look into DNA, and if you look onto this neuronal network, if we would replace these points here, I make it bigger. We have here kind of points. These points are representing raw data, actually. If we would put on their place the raw data, we will have exactly the same structure. We will have, again, A, T, C, and so on. Maybe not in this order, of course not in the same order, but actually the same. So we will, if we would have a big network with lot of layers, with a lot of neurons, we would have here huge combination, easily 3.2 billions actually. So it will be huge. And this is the reason why if you create a bigger network using some tool, PyTorch or TensorFlow or, whatever it is, it will be, if you create bigger network, it will be quite slow if you train it. So the bigger you create the network, the slower it gets because of huge number of parameters which are created. So huge number of combinations actually, and they are of big size. So you have big tables there. They are multiplied with each other and so on. We will learn it. As I said at the beginning, we will learn. We will have a dedicated session what is happening there under the hood. And this is the point, and this is the question to you. What do you think? Could it be that actually we say that our body is the result of kind of evolution or staged process? It looks to me like the DNA by itself is something like the product of a so-called neuronal network. It is, of course, it's not a neuronal network, but the idea is of combination, recombination, and combination again and again and again. It reminds a lot of things which are happening, how they are happening in nature. And I would be very happy if you would write a comment. And yes, so I wish you all the best. And next time we will work with similarities of neuronal nets, artificial neuronal nets, and real neural networks. See you next time. Write comment below. Don't forget like and subscribe. Bye.